Have you ever wondered what the future of agriculture holds in the face of growing population and changing climate conditions? The world is changing rapidly, and with it, our agricultural systems face immense challenges. As our population continues to skyrocket, expected to reach nearly 10 billion by the mid-century, the demand for food is growing exponentially. But it's not just about producing more food. It's about producing more nutritious, quality food, and doing so in a way that is sustainable for our planet. Agriculture, after all, isn't just about feeding us. It's about maintaining the delicate balance of our ecosystems, ensuring biodiversity, and preserving our planet for future generations. But with increasing environmental stressors, from rising temperatures to erratic weather patterns, our current agricultural practices are being pushed to their limits. Now imagine a world where crops are resistant to pests and diseases, where plants can withstand extreme weather conditions, and where we can grow more nutritious food on less land. It sounds like a dream, doesn't it? But it's not. It's a potential reality, thanks to a revolutionary scientific breakthrough. Food security is a pressing global issue, and it's not just about quantity, it's about quality too. Around the world, millions of people suffer from malnutrition, not because they don't have enough to eat, but because their food lacks essential nutrients. Addressing this problem requires a shift in our thinking, from simply growing more food to growing better food. In this complex scenario, the need for innovative solutions has never been so critical. As we venture into the future, we need tools that can help us navigate these challenges, tools that can revolutionize the way we grow our food. And so, we turn to science. We turn to a groundbreaking tool that has the potential to transform agriculture as we know it. A tool that can help us meet the growing food demand, improve nutritional quality, enhance crop resilience, and ensure a sustainable future for us all. Enter CRISPR, a remarkable tool that holds the potential to revolutionize our food system. But what exactly is CRISPR and how does it work? Let's journey into the world of microorganisms, where this amazing technology was first discovered. CRISPR, which stands for Clustered Regularly Interspaced Short Palindromic Repeats, is a natural immune system found in bacteria. It's their way of fighting off viruses, kind of like our body's immune system. Now let's understand this in simpler terms. Imagine you're a bacteria and a nasty virus invades your space. You fight it off, but not before copying a piece of its DNA. You store this DNA in your own genetic code as a wanted poster, so if the virus ever shows up again, you know exactly what to attack. That's what CRISPR does. It's a memory bank of viral villains. Scientists noticed this mechanism and thought, hey, what if we could use this system not just to recognize and destroy viral DNA, but to target any DNA sequence we want? And that's where the revolutionary idea of CRISPR as a gene editing tool was born. So, how does it work? Well, think of CRISPR as a GPS system that we can program with the coordinates of a specific gene. Guided by a molecule called RNA, CRISPR navigates through the complex genome until it finds exactly the gene we want to alter. Once there, an enzyme called Cas9, the molecular scissors, cuts the DNA at that precise location. The cell's natural repair machinery then kicks in to fix this break. We can take advantage of this repair process to introduce changes or edits to the genes. We can remove, add, or alter sections of the DNA sequence to achieve desired traits. For instance, in agriculture, we could edit the genes of a crop to make it more resistant to pests, diseases, or harsh weather conditions. This precision and efficiency is what makes CRISPR so groundbreaking. In essence, CRISPR can be likened to a pair of molecular scissors capable of cutting and pasting genes. It's a game changer, not just in the field of medicine or research, but also in agriculture, paving the way for a future where we could potentially engineer crops to feed our ever-growing planet. So how does this cutting-edge tool come into play in our fields and farms? You might be wondering. Well, the answer is as fascinating as it is complex. CRISPR's potential in agriculture is enormous, and it's already making waves. Let's start with crop yield. By subtly tweaking the genes of plants, we can enhance their productivity, making them more bountiful. Imagine a tomato plant that produces twice as many tomatoes, or a wheat field that yields an extra loaf of bread per square foot. That's the kind of game-changing difference CRISPR can make. But it's not just about quantity, it's also about quality. CRISPR can help us engineer crops with enhanced nutritional profiles. Picture an apple with twice the antioxidants, or rice that's packed with extra vitamin A. These are not mere fantasies, but real possibilities with gene editing. Now let's talk about pests and diseases. These are age-old enemies of farmers, causing widespread crop loss and food insecurity. With CRISPR, we can bolster the natural defenses of crops, making them more resistant to these threats. Imagine a world where the potato famine is a thing of history books, not a looming threat. Environmental stress is another major challenge in agriculture. Drought, heat, frost, and salinity can all wreak havoc on crops. But imagine if we could make our plants more resilient, able to thrive in a wider range of conditions. With CRISPR, we can do just that. We can engineer crops that can withstand harsh conditions, helping to ensure a steady food supply in the face of climate change. And the beauty of it all is that these changes are precise. We're not introducing foreign DNA from other organisms, as in traditional genetic modification. Instead, we're making small, targeted edits to the plant's own genetic code. It's like using a word processor's find and replace function, but for DNA. So the next time you bite into a crisp apple or enjoy a plate of rice, remember, science, specifically CRISPR, is at work. It's helping to ensure that we can continue to enjoy a diverse, nutritious, and abundant food supply. With CRISPR, we can tailor our crops to meet the demands of a growing world. So let's continue to explore, innovate, and harness the power of this incredible technology. Sounds promising, right? But like all great innovations, CRISPR in agriculture comes with its own set of benefits and challenges. Let's dive into the benefits first. CRISPR's potential to address food security issues is vast. By tweaking the genetic makeup of crops, we can make them more resistant to pests and diseases, reducing the reliance on chemical pesticides and contributing to a healthier planet. Imagine a world where crops can withstand harsh weather conditions. Thanks to CRISPR, that's not just a dream. Gene editing can help us create plants that are more resilient to environmental stress, such as drought or extreme heat. This is a game changer, especially for regions that struggle with adverse climatic conditions. Another significant advantage is the improvement of nutritional content. CRISPR allows us to enhance the nutritional value of crops, making our food healthier and more beneficial. This could be a giant leap towards eradicating malnutrition globally. Now, on to the challenges. One of the main hurdles is ethical concerns. While the idea of genetically editing our food might sound like a science fiction dream, it also raises questions. How far should we go in altering nature? And who gets to decide? Regulatory hurdles are another significant challenge. The laws around the use of gene editing technologies in agriculture are still in their infancy and vary widely across the globe. This lack of uniformity can slow down the adoption of CRISPR, as companies might be reluctant to invest in technologies that might not be approved in all markets. There are also concerns about the long-term effects of genetically edited crops on the environment and biodiversity. While CRISPR promises a lot of benefits, we need to be cautious and ensure that it doesn't inadvertently harm our ecosystem. Remember, every technological advancement comes with its set of challenges. The trick is to navigate these hurdles wisely and responsibly. While CRISPR holds great promise, it's clear that there are hurdles we need to overcome. We need to ensure that we use this powerful tool to create a better, healthier, and more sustainable future for all. So what does this all mean for the future of our food? Imagine a world where every crop is precisely engineered for optimal growth, taste, and nutrition. A world where farmers can grow food in harsh climates, where every plant is resilient to pests and diseases. This is the world that CRISPR gene editing technology is promising us. It's like having a master key to the genetic code of life. With this key, we can unlock the full potential of our crops. We can create plants that are more resistant to pests, reducing the need for harmful pesticides. We can engineer crops that can withstand droughts, floods, and extreme temperatures, ensuring a steady food supply even in the face of climate change. But it's not just about survival. CRISPR also allows us to enhance the nutritional value of our food. Imagine rice that's packed with vitamins, or wheat that's low in gluten for those with dietary restrictions. These are not just fanciful ideas, they are real possibilities that scientists are exploring right now. And let's not forget about the implications for sustainable agriculture. By creating crops that require less water, less land, and fewer chemicals, we can significantly reduce our environmental footprint. This is crucial as we strive to feed a growing population without depleting our planet's resources. Now, don't get too carried away. These are possibilities, not certainties. There are still many unknowns and potential risks associated with genetic modification. But with careful research and responsible use, CRISPR holds tremendous potential to transform our food system for the better. So, the next time you bite into an apple, just think, It could be a CRISPR apple, perfectly crisp, sweet, and genetically designed for your enjoyment. Now, isn't that a tasty thought? With CRISPR, the future of food is not just a dream but a possibility within our grasp. Don't forget to subscribe to Brainiacata for more enlightening videos like this one. Before we wrap up our exploration of CRISPR and its potential impact on agriculture and our food system, let's take a quick detour to understand how we got here. Gene editing, the process of manipulating an organism's DNA, has been around for decades. It all began with early techniques like selective breeding and cross-pollination, which farmers have used for thousands of years to improve crop yield and quality. But these methods were time-consuming and imprecise, and it was not until the 20th century that scientists began to develop more sophisticated tools for genetic manipulation. Enter recombinant DNA technology, a method that allowed scientists to cut and paste genes from one organism into another. This was a game changer, enabling the creation of genetically modified organisms, or GMOs. But even then, the process was complex, costly, and somewhat hit or miss. Fast forward to the 21st century and we have CRISPR, a tool that is more precise, more affordable, and more accessible than any of its predecessors. It's like going from a pair of scissors to a molecular scalpel, allowing us to edit genes with unprecedented accuracy. So, as we stand on the brink of a gene editing revolution, it's important to remember that this is not just about CRISPR. It's about the culmination of centuries of human ingenuity and scientific progress, a journey that has brought us to the cusp of a new era in agriculture and food production. And who knows what the future holds? With the pace of scientific advancement, the next big breakthrough could be just around the corner. Stay tuned to Brainiacata as we continue to explore these exciting developments. Don't forget to like and subscribe for more fascinating insights into the world of science. In this segment, we're going to delve a little deeper into the intricacies of CRISPR and how it works at a molecular level. If you think back to your high school biology classes, you'll remember that DNA is made up of a sequence of 4 bases: adenine, A, cytosine, C, guanine, G, and thymine, T. These bases are the building blocks of life, and their precise sequence determines everything from your eye color to your susceptibility to certain diseases. CRISPR, which stands for Clustered Regularly Interspaced Short Palindromic Repeats, is a system that bacteria use to defend against viral attacks. The bacteria capture snippets of DNA from invading viruses and use them to create DNA segments known as CRISPR arrays. These arrays allow the bacteria to remember the viruses, and if they attack again, the bacteria produce RNA segments from the CRISPR arrays to target the virus's DNA. The system also involves a set of CRISPR-associated proteins, or Cas proteins. The one we're most interested in is Cas9, which acts like a pair of molecular scissors. Guided by the RNA segments, Cas9 can find the invading DNA and cut it at the exact point required. Scientists realized that they could harness this system and program the RNA to target any DNA sequence, allowing them to edit genes with incredible precision. So, in a nutshell, CRISPR-Cas9 is a tool that uses RNA molecules as guides to target specific DNA sequences in an organism's genome, and then uses the Cas9 protein to cut the DNA at the targeted location, allowing scientists to add, remove, or alter specific genes. The possibilities are truly astounding, and we're only just beginning to scratch the surface of what we can achieve with this powerful technology. Up next, we'll explore some of the potential applications and implications of CRISPR in agriculture. But before we do, don't forget to hit the like button and subscribe to our channel for more deep dives into the world of science. Welcome back, science enthusiasts! Now that we've delved into the fascinating mechanics of CRISPR, we're going to shift focus a little bit and discuss some of the ethical implications of this powerful technology, particularly in the realm of agriculture. As we've discussed, CRISPR allows us to alter the genetic makeup of organisms with unprecedented precision. This opens up a world of possibilities, from creating crops that are more resistant to pests and disease, to engineering animals that grow faster or produce more milk. However, these advancements don't come without controversy. Some critics argue that we're playing God, meddling with nature in ways that could have unforeseen consequences. There are concerns about the potential for creating new diseases or upsetting ecological balances. On the other hand, supporters argue that CRISPR could help us solve some of the world's most pressing problems, like hunger and disease. They point out that humans have been selectively breeding plants and animals for thousands of years, and that CRISPR is simply a more efficient way of doing the same thing. As we move forward, It will be crucial to have an open and informed debate about these issues, balancing the potential benefits with the ethical considerations. As always, we'd love to hear your thoughts on this. Comment below with your perspectives on the ethical implications of CRISPR, and don't forget to subscribe for more insightful discussions on the latest scientific advancements. Welcome back to our deep dive into CRISPR, folks. After weighing the ethical considerations in the previous scene, let's now explore some real-world applications of CRISPR in agriculture through a few case studies. Firstly, we have the case of the Arctic apple. This apple variety doesn't brown when cut or bruised, all thanks to the magic of CRISPR. Scientists used this technology to turn off the gene responsible for producing the enzyme that causes browning, thereby extending the apple's shelf life. Next, we have the example of a genetically modified mushroom. Thanks to CRISPR, this mushroom doesn't bruise or brown easily, thus reducing food waste and increasing its appeal to consumers. Finally, let's look at how CRISPR is making strides in livestock breeding. Scientists have successfully used this technology to engineer pigs that are resistant to a deadly virus, potentially saving millions in the pork industry while also improving animal welfare. These case studies illustrate the immense potential of CRISPR in revolutionizing agriculture. However, as we've discussed earlier, it's crucial to balance this potential with ethical considerations. As always, we encourage you to share your thoughts. Do you think the benefits outweigh the risks? Are there other examples you'd like us to explore? Comment below and don't forget to hit the subscribe button for more engaging content on cutting-edge science. Thank you for joining us on this journey through the world of CRISPR and its implications in agriculture. We've delved into the science behind this revolutionary technology, explored its potential benefits and challenges, and even took a peek at some real-world applications. We've seen how CRISPR can transform our food supply, from creating apples that don't brown to engineering virus-resistant pigs. But as we've also discussed, these advancements come with their own set of ethical considerations. As we continue to push the boundaries of what's possible with CRISPR, it's essential that we ask ourselves, how can we ensure that this technology is used responsibly? How can we balance the potential for innovation with the need for careful oversight and regulation? These are questions that will continue to shape the future of CRISPR and agriculture. As we wrap up, we invite you to continue this conversation. Share your thoughts, engage with others, and stay curious. Together, we can navigate the future of agriculture and CRISPR. Stay tuned for more intriguing topics and don't forget to like, share, and subscribe. Until next time, keep exploring.